this usually continues with sometimes it continues with the previous discussion as well. To the student participant, I request that you should share the link of this roundtable meeting to as many fellow students as you can, because MBSI is organizing such events to share knowledge, and students will be getting benefited if they are working, maybe PhD scholars or any researcher who are doing some work. They can also share their experience or troubles that they are getting during their research. So, without wasting much time, I think we should start. We have our uh, president of MBSI, Professor Arvind sir, Dr. Prakash Salami sir, Dr. Arvind, Dr. Richa ma'am, Dr. Sandeep sir, Dr. Mobi Sara, and all other participants. Thank you for joining in. Uh, we we can start discussion about the topic of microbial polymers. So before beginning, I would like to ask if somebody already know about it or somebody who has any idea about or working already on some microbial polymers. Dr. Richa ma'am, are you there?
Hello, good evening, sir. Yes, I'm there.
Yes, ma'am. Please tell something about your work as well as if it is related to microbial polymers. Please share
No,
your
no, sir.
experience.
My work area was actually different. It was on biopulping. So, out
Okay.
of curiosity, I have joined this just to know something about that.
Okay. Biopulping, you said
Yes, yes.
it is related to uh,
Pulp
cellulose
and paper.
removal, pulp and paper industry. Yes.
Yes, sir. Yes. So, I had completed my PhD from Forest Research Institute, Dehradun, in the year
Okay.
2012. Yes.
Okay, great. So I hope you are also aware about bacterial cellulose.
Sir, actually, I had taken some gaps, so I'm rejoining the things again.
No problem, no problem. Just because we are going to discuss about all the different types of microbial
Okay.
polymers, and
Yes.
among them is bacterial cellulose.
Yes, of
So
course, of course.
cellulose
Yes, sir.
usually we get from plant sources
Yes,
in
sir.
large quantities, but
Yes.
some
That is
bacteria
used.
is also
That is used in
synthesized.
yes, yes, in making paper and all. Yes.
Yes, yes, and because it is bacterial origin, and so they are as good as cellulose. Their structure is a bit different, but they can be used even in medical uh, application.
Okay.
So
Okay.
bacterial cellulose people are preparing to avoid the burden that we are giving to nature.
Yes. Mm-hmm. Okay.
We are using a lot of plants. No? We are growing plants or using agricultural land to grow such things, and then we use a lot of reagent to get the cellulose out of that.
Yes, sir. Hmm.
So bleaching agent is needed and then it goes in the effluent. Although there are mandates of almost zero discharge plus environmental policy is also there.
Yes,
But
sir. So
we
that is.
do use a lot of chemical, no?
Yes, because of that only this biopulping option we worked on. But on ground level, it is not possible to scale up these things. So that's why it's not too much in trend. So this is a good thing which you are telling that cellulose from bacteria. That is a very interesting topic. So
Yes, yes, and quite relevant to yours or maybe similar.
yes,
Uh,
yes. Mm.
yeah. Can you shed some light on this biopulping? This is a new term, biopulping to me.
Sir, actually, things which we are using, major chemicals which we are using in preparing these pulp from the wood for making the paper, that is creating a lot of environmental hazard. So that's why we worked on two fungus, that was Schizophyllum commune and Trematis versicolor. And I just took this eucalyptus chips from Saranpur, and these two fungus were just grown on these chips, and during a period of time, on various parameters, changing various parameters, we checked like in what time it is degrading the lignin of the wood, so that we can extract the cellulose which we are otherwise extracting by using these chemicals. So it would be beneficial if it can be done with the help of these microbes, fungus. But because these conditions we cannot, in the microbes, what happened, the conditions we cannot maintain in the higher scale. So these are actually giving results on the lab level, but if we are scaling it, they are not giving that much. So what I did in my PhD, that up to that level, the results were positive, but I cannot say what will happen if it is applied to large scale. So this is what
Yes, most of the biological process is facing such issue,
yes, yeah,
but obviously, if once it will get successful,
mhm,
it would be helpful to nature.
yes, sir. Mhm.
Mm, Great, glad to know.
Thank you.
Any other participant? They want to speak. Dr. Mobi, are you there?
Hello, sir. So
Yes,
myself is Dr. Ganga. We
yes.
have find some organisms which is producing a large amount of gummy-like structure. I mean, things in the possibly it is EPS, but we tried it with cellulose and FTR. It is not found as cellulose. So, what could be the possible one which we can expect in that scenario?
Your voice was breaking. So far, I could understood is your organism is secreting something which you are expecting, like slimy kind of thing.
Sir, I
You
am in a
want to
hospital. That's
characterize.
what.
Uh-huh.
Veena, my student is there. Will
Okay,
you
okay.
respond? Veena?
Hello,
Yes, Vina.
ma'am. Am I audible? Sir,
Yes,
am
you
I
are audible.
audible?
Yes.
Sir, we are trying to extract, synthesize cellulose from bacteria, but we got a slimy, like a hydrogel-like substance from the medium, and we analyzed that material and we dried that into thin films and analyzed by FTIR, and from that we find out that it's not bacterial nanocellulose. Then what will be the probable chance?
Okay. No. So, is it kind of pellicle structure which is happening in the culture at the air liquid interface?
No, sir, not at the air liquid interface. It's at the bottom. It's a thick hydrogel like substance, like EPS, but it's
Mm-hmm.
too thick. Almost in a beaker, it's almost 5-6 centimeter thick and
Think, within 72 hours, or how long does it take?
sir, it's up to 10 days of incubation.
Okay.
10,
Uh,
11 days of
Yes,
incubation.
yes. So, to check if something is kind of carbohydrate structure, we can go for this MS. Okay, we can go for MS, but we need to derivatize the glucose unit so that we can make them mobile in the means chromatographic phase earlier. Okay. And if you
So,
suspect that it is not a carbohydrate, it may be a, although most likely it would be carbohydrate, okay, because it is an
yes, sir.
extracellular substance. Yes.
Mhm.
So I hope you know that there are, you know the organism which you are working on.
It's a Bacillus
Bachelor only,
species.
sir.
Okay. So, because some organisms are already known to secrete polymers, in this case, in your case, it may be the secretion would be too, means secretion is in large quantities, right? Bacillus also
Yes,
produces EPS. Yeah.
yes.
So, in order to characterize it, I think FTR is just qualitative analysis. So, better go for MS.
Okay, sir.
Mhm.
Thank you, sir.
And sir, if it is EPs also, since it is very thick, can you say any kind of application, possible application in this context?
Yes. So possible application of EPS, it depends on the quality of the EPS. Okay, what kind of monomeric units are there? But various microbial polymers are already like Pululan is used as a like mouth freshener strip, na? So there it is just like chewing gum. So chewing gum has something else, but Pululan coated with sugar material can be used as a mouth freshener. Okay, sugar plus freshener, mouth freshener component. So that will act as a biodegradable thing and it don't hamper anything once it gets to the gut because it is a biodegradable polymer and biocompatible as well. In addition to that, there are several biomedical applications which may be explored. For example, other polymers like polyhydroxyalkanoate, uh, they are used in biosensing application, drug delivery, and this suture preparation of sutures, no, biodegradable sutures, even heart valves. So there are several applications of microbial polymers, but it depends on the quality and their compatibility with the thing that you want to use, especially in the medical field. It must be biocompatible, it should not be immunogenic. So if this polymer is containing any kind of protein also, then it will be act as a foreign and the body may reject or start showing immune reaction. Right. So first we need to characterize the polymer and the potential application could be many things. Simplest one would be maybe if it is a cellulose, it can be used for paper making or for making devices, biodegradable devices. Okay, in biosensor application. If it is just EPS, it can be used to treat how to say in wastewater treatment plants. Such things are needed to make a lot of secretion where they can these other microbes and pollutants can get attracted to this polymer and they get settled down and means they make flocule, na? So flocculant, as a flocculant it can be used in wastewater treatment plant. So there can be various application. Uh, if anybody else have some idea or suggestion, they may also give their input. So far, I understand I have already provided some of them, but maybe Dr. Sandeep, are you there? Okay, Dr. Arul sir has also joined today.
Good evening, sir.
Yes, yes, Sarika ma'am.
Uh, I'm Sarika, PhD scholar, sir.
Okay, ji. Yes, tell me.
EPS production. So I just want to ask Ganga ma'am, like which medium she is using in this in her process.
Ma'am, we are
That they can.
using HS medium, Hestrin
Which
Shram
medium?
medium, Hestrin Shram medium.
Is it containing the salts only?
No, ma'am, it's a sucrose containing medium.
Okay. So maybe because of the sucrose you are using is medium, it is production in the EPS. As I'm working on the same EPS production, and I have like noticed when I'm using sucrose and the carbohydrate external source in the medium of the bacteria, there is very little amount of that sticky amount that you said is produced, or like it in negligible amount it is produced. Like in my work, I generally combine the protist and the bacteria. In that, I'm getting the more amount of exopolysaccharides, while in the only bacterial medium in which I have not added anything like just the simple mineral salt medium, I'm getting very negligible amount of sucrose. EPS, sorry.
EPS, yeah.
Yeah. So maybe because of that extra like carbohydrate source we are providing in the medium, it is producing EPS like.
Hello.
Yes.
Yes, yes. So, yes, you are right. Actually, to produce EPS, we need carbohydrate or carbon sources so that they can be
Yeah.
produced in large quantities. And also, it is the ability of the bacteria because not all the bacteria can synthesize as much quantity as they were saying. So, it is quite good observation they have, and they are looking for some kind of solution. It seems like application oriented. So,
Yeah,
if
because in
they
my process, like I am using the consortium, like two microbes, protist and the bacteria. So because of the protist ability of grazing on the bacteria, it is like acting as an ecological stressor. And by because of that stress, the bacteria is producing EPS in very large and enhanced amount without any addition of the carbohydrate like external source.
okay, okay.
Yes.
But how they are getting that is good. This is a new thing to me also. But uh, maybe they have to synthesize a lot of carbohydrate, no? So they need some kind of carbon source or energy source to produce it.
Yeah, it needed, but in my medium, generally I use the pH saline medium or the nutrient broth normally and the soil extract. Only these materials I'm using in the medium, and then the bacteria and the protist, and I'm getting the enhanced amount of EPS.
Good, good, good.
Yes.
So, uh, and you have already characterized that EPS. No, so characterization also you can emphasize the a bit so that they are also willing to characterize. No, so if it is EPS, how can they characterize?
Yes. Like
So
one
are
problem
you?
I'm getting in this, like generally we use the extraction process by using the isopropanol, the alcohol, and it uses like in large amount, like 2x or 3x we use generally in the extraction. So I just want to remove this step of the isopropanol. So any suggestion to use the other processes for the EPS extraction?
Isopropanol or acetone, so far I know these two are commonly used.
Yeah, but
Isopropanol
these
or
liquids are
acetone.
used in huge amount, no? Like double or triple we are using. So it becomes some costlier process.
Acetone is not that costly. Isopropanol is also, I think, even any kind of alcohol, like maybe, but acetone is considered as green solvent compared to other expensive solvent, and cost-wise also. Uh,
So any
otherwise,
other
uh,
processes?
no, aqueous two-phase extraction, if it is possible, we need to check with the solvent and the solubility. No, I have not worked on EPS, so I cannot tell. But maybe if some other participant, those who have worked, they may shed some light. Dr. Sandeep, are you there? Anyway, I think KBS Lakshmi want to say something. KBS Lakshmi, are you there?
Sir, I have a doubt whether it is acetone or isopropyl alcohol. If it is in the cold condition or we have to use in the room temperature condition.
So acetone you can use in cold condition. Na? Isopropanol I have not used, so
No, sir.
I think.
Actually, in both we use the cold temperature for isopropanol
Hmm,
also and the acetone. I generally use the isopropanol for my project work.
okay, okay. So cold acetone method I have read and followed one time only. But that's good. All the solvent that we use is usually chilled, so chilled ethanol or chilled. acetone can be used.
Sir,
Yes,
uh, so by just doing the centrifugation like two, 3 times of the medium, can we get the pure form of the EPS?
pure form we cannot get because once you are centrifuging, means we need to remove the cells. No, we need to, you want to purify that, right? So purification will happen when we will remove all other biomaterials. So we need to remove lipid as well as proteins. Then
Okay,
only
like for
EPS
that
can be purified, no?
protein. Yeah,
So you
yes,
can use proteinase, but then it will become expensive, no? Enzymatic method.
yes,
Maybe you can use immobilized enzymes that can be recycled, but it will not degrade all the type of proteins. Some proteins means there are category of proteases, right? So
yes.
not all the protease will work in this case. And then you are also left with lipid. Some of the lipid may be soluble in acetone as well, even ethanol.
Yes.
So minor quantity is there, but we need to. That's why you whenever you see some downstreaming application, very often it is said that if you reduce the number of step, then cost goes down, but purity we have to sacrifice or compromise somewhat towards the purity.
Okay.
The finest purity, like 90% or above, if you want, then we have to become very specific or we need to increase the steps of purification.
Yes. Sir,
Mm.
actually, one process which I have made, like first will actually I am getting the EPS in both the flask medium, like liquid medium, and also I'm getting in the plate, like normal petri plate in that agar medium. I'm getting the pure form of EPS, like slimy like structure.
Yes, but that slime will also contain bacteria, na?
Yeah, so we can done the like heat the EPS like 65 to 70 degrees Celsius for one time, then centrifuge, then heat, then centrifuge.
So
Will it work?
you can essentially kill the cell, but the protein component will remain there, na? Protein as well as lipid component will remain there. So yes,
Okay,
you
for
can.
that we can do the enzymatic cleaning also, no proteinase K or DNase or RNase.
Yes, because EPS may or may not include nucleic acid, but there can be other like bacterial enzymes will also be present, extracellular enzymes embedded
Yes,
within the EPS, and
yes,
then comes the main polysaccharide part also. Because you
yes,
said purification, so I'm saying that one you want to purify. The pure form means we it should be devoid of all other biomolecule. Right. When you are going to characterize it, or suppose you want to use it for some biomedical application, then it won't be suitable. But for all other applications, like you want to make some kind of drug delivery agent, nanoparticle, or anything like that, and then drug delivery also it may be having some extra protein. So for drug delivery you cannot use, but for environmental applications, flocculation, those kind of work, you can use it. And then
okay,
purity
so
may not matter. Mm.
okay. So by just heating centrifuge and the enzymatic cleaning, I will get the pure form of the EPS, or still I will not.
So that's what I'm saying. Na, what you want with the means what you understand by the pure form of APS. Is it just devoid of cells? Then your process is correct and it is reasonable. Okay, it will be
Okay,
devoid of cells and it will won't allow any further growth because you have heat killed the organism. Okay.
okay,
Hmm. And also remember that those who organism which can make spores. So if your organism is bacillus, which can make spores, then heat killing will not work alone.
oh,
No? Yes, and proteases can work on some proteins. So you need to define or use different types of protease,
yeah,
sorry, different types of proteases. So optimization has to be there to check for the purity or whether some protein is still remaining or not.
So the purification part will be done by using the dialysis or the column chromatography, I guess.
Yes, dialysis is also fine, but I'm telling you, na, so you have increased a step, and dialysis is not very cheaper. When you need some membrane and a membrane cutoff, molecular weight based separation happens there. So the polymer usually have a large molecular weight, so it will allow all other smaller particle also. So it will be a kind of exclusion where you can keep the polymer within the bag, so within the dialysis bag, while the smaller molecule may float around, so they can go away from the bag. But it will be a bit expensive if you consider large scale. So maybe
Yes.
microfiltration type of thing may be used. Ultrafiltration, microfiltration, there are membranes, no? For industrial scale, there are membranes that people use, and even tangential flow filtration may also be tried because that is quite cheaper. Tangential flow filtration, TFF.
Oh, thank you, sir.
Thank you so much. Uh, Anyone else? So I think we started talking about research, but just let me come back to the main theme, that is microbial polysaccharide. They can be of various types. To this, I am this new statement that I am going to give now is for those who are very new to this area and they want to get some information. So to them, I would like to tell that there are several polymers of bacterial origin. Some are quite common that you may have heard of, like xanthan, dextran, pullulan. Okay, they have been uh, worked out since maybe past 60 years or so. Okay, so we know that there are extracellular or capsule, which is very much made up of polysaccharides secreted by the bacteria itself. Right. So there are several category of microbial polymers in which polysaccharides are um, capturing the large part. But there are natural biopolyesters like polyhydroxyalkanoids that can also be produced by bacteria. Then there are some compounds, some polymers that can be synthesized with the help of bacteria like polycaprolactone or polylactic acid. Okay. And then there are polyphosphates also. They also come under polymer category. So there are several different types of polymers that bacteria produces. Out of them, these polysaccharides like EPS we were discussing, or so they are why they are produced. Sometimes they are produced as a storage material like we also keep glycogen in our body or liver, so we store excess of carbon, right? Excess of carbon can be stored within bacteria in the form of glycogen, inulin, or polyhydroxyalkanide, right? And in other cases, bacteria or microbes secrete these polysaccharides in the form of xanthan, dextran, levans, etc. So they are considered as extracellular polymeric substances or sometimes extracellular polysaccharides. Okay. Abbreviation form will come as EPS only. Okay. So if we are considering extracellular polymeric substances, then that may or may not contain nucleic acids as well as other material. But once we are saying it is extracellular polysaccharide, then it is a polysaccharide only. While if you can categorize, they can be involved in capsule formation, sheath formation, or just like a biofilm. So people are working on biofilm research both in wastewater treatment as well as in the clinical setup. People want to work on biofilm. For clinical aspect, people want to degrade biofilm, so they want to degrade EPS, while in the wastewater treatment plant, they are willing to form as much of biofilm as they can so that clarification gets easier and there could be a lot of heavy metal removal as well as other pollutant removal may happen by the action of microbes. So this was a brief introduction of the topic on microbial polysaccharide. Now I would like some other participant who are willing to discuss something or they want to uh, tell about their research on biopolymers. Asmita, Asmita, are you there? Asmita Bambole, Dr. Sandeep. Last time I missed you. Uh, I think you were online only. Dr. Sandeep, are you there now? Dr. Sandeep, Dr. Kalidos.
Yeah, tell me, sir.
Yes. Can you tell us something about yourself and your research work? Is it related to microbial polymer?
No, I completed my work in exopolysaccharide during the year 2004. During my MSc work, it was in xanthan gum. I
Okay.
extracted xanthan gum from the bacteria Xanthomonas campestris. Okay,
Okay.
how do I isolate the bacteria? Let me explain it. I isolated the bacteria from black rot, cabbage, and cauliflower. I collected them from market. Okay, and I extracted. I Yes, I scrapped only the black rot affected portions and serially diluted it and plated it on nutrient agar media. And
Mhm.
later I got yellow color pigment producing colonies. Okay, and identified those yellow color pigment producing colonies as xanthan gum producing.
Okay.
Then later on, I went for extraction of xanthan gum, and that paper was published in Indian Journal of Applied Microbiology.
Okay.
Yeah, that is my
Great.
that is my own MSc work.
Hmm.
And now I'm working as an assistant professor in the Department of Microbiology, SRM College of Arts and Science, here in Chennai, sir.
Okay, great. You are working at SRM. My friends are also there. Uh,
Yeah.
glad to know that you have worked on this. So it was your master's project.
It was my master's project years back. It's, during 2004, I worked it.
Yes, yes. Then you can shed just minor light about xanthan gum extraction. So I hope the audience is clear that it is the cabbage and some plant that gets infected by Xanthomonas species.
As it is one of the phytopathogens, we can extract the Xanthomonas campestris from black rot disease of cabbage and black rot affected cauliflower. You have to collect
Mhm.
it and you have to serially dilute it and you have to go for the isolation. By using nutrient agar, we can do the isolation.
Yes, yes. And it is called gum. So have you extracted or have you noticed? Is it like a jelly kind of substance or what when you extract it?
It was like semi-solid consistency. It was semi-solid consistency. I have gone through. It required. I prepared some liquid media.
Hmm.
Liquid media, and from that liquid media in the sense broth culture. And from the broth culture, after a series of processes, we added double the volume of ethanol. Either ethanol or methanol should be added. There is a specific methodology you have to go through that. These are things still I remember. I too have to go through the paper right now.
No problem, sir. Just for the participants, they will get the flavor because I have not worked on it, and it is great that you have this experience already. So,
Yes,
yeah, because in our participants, no, there may be some research scholar, there may be some
yes.
students,
That,
master's
that is why
students.
I
No?
wanted really to join this discussion today.
Yes, yes, and we also get some new information. Like previously, we were discussing with Sarika, and I think Sarika and someone else, Geeta ma'am or somebody. So, so this way we also get some kind of clues, or sometimes we get puzzled, but we also gain knowledge here. Thank you so much.
Yeah, thank you. Welcome, sir.
Mm.
Welcome.
Yes, yes. So, any other participant who would like to speak? Mahadev Roy, are you there? Mahadev Roy, Muhammad Abdul Rahman, Muhammad Abdul Rahman, are you there? Prabhu, I think
Yes,
you
sir.
have been. Yes, Rahman, yes. Uh, so, are you a student or working, means doing some kind of research on?
Sir, I'm a student, sir, of BTech Biotech second year.
Okay, okay, great. So, you are doing some kind of project-based learning during your BTech?
No, sir, not started yet.
Okay, okay. I think from third semester onwards, uh, people, mean some of the colleges, there, universities, they're running this program, project-based learning, so that you
Yes, sir.
can get while reading, you can also get the information, means how to do research, or through research you can learn what you are reading in the class.
Yes, sir.
So we are very glad that you have joined in. This also motivates us that okay, students are listening and they are also learning, and somewhat we are helping them to understand on the topic. Okay, great. Professor Iqbal has just joined. Professor Iqbal, are you there?
Yes, sir. Yes, sir.
Professor, so glad to see you, sir.
It's my pleasure. I just
Yes,
seen
sir.
that there is some uh, meeting going on, so I thought that let us join and see
Yes,
what
sir.
is okay.
Yes, today Vipran sir is not there, and you and Professor Halami is already there, means in this meeting, and there are several participants. So now we are covering the topic of today is microbial polyester, sir.
Oh, that's fine. That's fine.
Yes.
Very nice.
So microbial polymers, including xanthan, fullulan. So most of the participants are working on EPS, while my expertise is on only on PHA. Largely, I have worked on polyhydroxyalkanoids.
Oh, that's fine. It's very
Yes,
interesting
sir.
topic to work. Yeah.
Yes, sir. So I
So
would
your
like, sir.
talk is over.
Uh,
Your talk is over.
no, sir. It
Your
is like
talk is over.
sir. It is like a roundtable discussion. No, so we are allowing all
Oh,
other participants also to speak. Otherwise, it could become monotonous also. Yes, yes. So that's why I also called you
ah,
to share so that you will share your knowledge. And
okay.
I'm
Okay.
surprised that today miss after a long time I'm seeing you. We have never met in person, but I know you through your Google Scholar and other sources.
Thank you.
Yes, sir.
Thank you. Thank you.
Yes, sir. So please shed some light on any kind of microbial polymers if you know of or you have been working on. No.
Basically, I have not much experience on working microbial polymer. But as a teacher, I used to teach students this topic in industrial microbial technology paper. So what are the different types of the polymers that we can get it extracted from through the various microbes utilization. So this is of course an topic of interest for me also. Uh, but exactly I'm not working on this, isn't it? So because
Okay.
some work I do on biofilm. So most of the students they are just concentrating on the EPS that
EPS,
is
yes.
produced during the biofilm formation. But actually, the microbial polysaccharides are polymers which are important. We are not covering in our research point. But these are
Mm-hmm.
the good work. If any students or any one is interested in doing, I appreciate that we can give the topic to our master research students also to explore different types of
Yes, sir.
polymers. Uh,
Especially even like degradation of this EPS for biofilm, we are targeting EPS means so that
yeah,
it won't form, right?
yeah,
While wastewater in wastewater plant, people need biofilm, so they want the biofilm to be produced and in large quantities rather.
yes, 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 yes. So the
Yeah.
enzymatic approach is there for degradation of the EPS
Mm-hmm.
matrix of the biofilm. So that aspect can be explored. That what maybe the enzymatic combinations or what are the enzymes that can be extracted from the microbes to be uh, used against the biofilm EPS matrix dissolve it to prevent the biofouling or the clogging, as you said. So these aspects are interesting, and
Mm-hmm.
if you are interested in this area, that's good. We appreciate that you are working in this area.
Yes, sir. Here nowadays I have started working on the antibiofilm activity of some natural compounds. So we are
That
working on
that
indole.
natural compounds you obtain from microbes or you are
No,
other
plants,
sources.
plants, sir. Plants
Okay, okay,
and
okay.
fungus also. Yes.
That's good, good. So there is some common interest is coming, between you and me.
I told you, sir, I know you through whenever I search, na, I see your name also. And some mutual connection is also there, but I'm forgetting how. Yes, sir.
So you can we can discuss separately if you have any
Yes, and
other
maybe next
discussion.
next roundtable discussion we may keep on biofilm alone.
Yes, yes,
No,
yes.
if
That
it
that
has
will be
not been
good.
done, yeah, we'll
Yes.
see to it.
So I will also participate actively if you plan for it this. Na?
Sure, sure, sir.
Okay.
Thank you
That's
so much,
fine.
sir,
Thank
for
you. Thank
joining.
you.
Mm,
Okay.
yes. Participant, anyone else who would like to speak something? Uh, Sheila, are you there? Sheila Joshi? Sheila Joshi, are you there? We also have Mahadev Roy. Mahadev Roy, are you there? Reshma Ramananda, any one of you, if you would like to share something? Thank you, Ganga. Thank you so much for the appreciation. Without your or all the participants actively engagement, this is not this won't happen. So that's good that you all have joined in. And although we had good discussion already, but we'd like to have more information from the participant side as well. Uh, so Sheetal Simran, Sheetal, are you there? Sheetal Gola. Then there is some Xiaomi. Someone has joined using that phone. Uh, Maybe they can introduce. Any participant can speak on the topic. It is quite interesting, and a lot of different polymers are there of microbial origin. So if not, I would.
Sir,
Yes,
so I have a
Sarika.
question from Sir Kalidos, sir, that he work on the Xanthomonas bacteria. So actually, the Xanthomonas bacteria is a plant pathogen, and its colony formation is like yellow sticky colonies that formed in the plates. So how can you distinguish like that the yellow color compound, sticky compound, is only the EPS, not the bacterial biomass, because both of them looks like same yellow color slimy only. So what are the techniques you used in this method? Like what analytical techniques you use?
There are specific tests for identification of xanthan gum. You can go through that. But as of now, I don't remember. I worked it during 2004. Xanthan gum extraction and identification of xanthan gum and production of xanthan gum. I worked it during 2004, and the paper got published during 2005. And there are methods available, simple methods for confirmation of xanthan gum. It is available. We can adapt any one of the methods.
Okay, thank you, sir. Actually, I just confused. Like, I'm also working the antimicrobial activity by using the Xanthomonas bacteria, and it is same like you said, the yellow color producing. So I thought, is it EPS or that colony only? So
Okay, we have to go through simple. There are simple biochemical tests available to check whether the produced yellow color pigment is xanthan or not. We can confirm it.
Okay, great. Shivani Tiwari has recently joined. Shivani Tiwari, are you there? Shivani? Shivani, Simran,
Yes,
yes. Yes, tell us something about yourself, whether you are a student or a researcher, and are you working on microbial polymers? Yes, Shivani, over to you. Shivani, are you there? Okay, so finally I call upon Professor Prakash Salami sir. Salami sir, are you there?
Yeah, I am very much available.
Yes, sir. I did not wanted to disturb you more, but I think now your useful insight is needed here. I hope you have heard some of the discussion. Maybe you'd
Exactly, it
like
is
to add
going
upon
on in
something.
a quite right direction, and then we could see research scholars they are explaining the difficulties which they are facing with reference to whether it is a exopolysaccharide or cellulose, right? We in
Yes,
fact
sir.
worked on the EPS from Bifidobacteria. We have that paper in Journal of Applied Microbiology where we have reported the heteropolymer production with the specific moieties of fucose sugar, which was not demonstrated before. And you know that
Okay.
the CPS actually exhibit a prebiotic activity, you know.
Yes.
Yeah, most of this actually researchers they forget that when we look for this polysaccharide production, what should be the cultivation media? So we always see CN ratio, glucose to nitrogen, and
Mm-hmm.
we actually have to see that glucose is more 5% or 6%, you know. One of the major difficulty, whether it is a EPS or the cellulose or even the, you know, I think you worked on the PHA, polyhydroxy.
PHA, yes, sir.
You know, because the microbial processes wants to be the expensive because of the media conditions or the media requirement. And this is actually true in case of the CPS even. because we have to actually feed the microorganism with lot of nutrient-rich media and wait longer
Mm.
time.
Yes,
In
sir.
EPS, PHA, what happens is the duration of cultivation, like alkaligens latus, 80
Hmm.
hours you have to wait, right? Bacillus in 30 hours. And then you just
Yes.
get milligram per liter, which is actually very.
Hmm.
And you as a PHA is a intracellular. So breaking,
Yes, sir.
you have to actually
Extraction is a bit difficult. Yes.
organic solvent you have to use, and then much more cost intensive process.
Yes, sir.
People actually have made some attempts with reference to looking some alternative methods, biological processes, enzyme hydrolysis, and extracting the PHA.
Yes, with PHA there are so many things, sir. Actually, I thought to first to cover the because my expertise lies there. No, I have been working in this area for the past I think since my PhD. So I
Exactly.
have still not left this. But yes, you are
Yes.
very right that the production of PHA in large quantities is quite difficult.
Exactly.
And then, yes, but there are several novel things that people have tried in Malaysia, especially. I would like, for the participant point of view, I'm telling this that what they did, they made a pellet, they produced the intercellular PHA in an organism, recombinant organism, maybe alkaligenous strain only. Okay, then they dried the biomass in the form of pellet and they gave to mouse and mealworm and some others. Okay, so that they will eat that. Their digestive enzymes will eat all the proteinous material, but there is no enzyme to degrade PHA. So the final fecal material will contain a lot of PHA that can be segregated by washing alone.
Exactly.
So such kind of process they develop, although how commercial this is, that I don't have an idea, but they have published a lot of paper using animal models to extract the polymer because it is intracellular polymer and we need to use organic solvent. So to avoid such thing, they are feeding the bacteria to a live organism and collecting pellets and purifying the polymer.
Exactly. So economically, it is not feasible process from the
Yes.
microorganisms. But the thing is, we have to one other day have to depend on the microorganisms. Now, several recombinant strains they have developed. Even we worked on that. Cheaper carbon sources people have used, like we use actually moha flower juice extract. You know,
Hmm.
that
Hmm.
actually gives a copolymer also.
Yes,
Polyhydroxyate
sir.
to valerate. Very
Yes,
important.
yes.
We have some publication from that. Yeah.
Okay.
And then even we use actually starch as a carbon source. And
Uh-huh.
bacillus, because bacillus actually grows much more faster, not like alkaligens latus.
Hmm.
So this is how actually are there, or one can actually look for the solution when
Yes, sir.
we look
Uh,
this PHA
I
product.
Yes, sir. If I heard you correctly, you said you used mahua uh, plant extract or some carbon source
Mawa flower
of flowers.
juice. Mawa,
Oh, great,
yes.
great.
Because
See,
rich
recently,
in sugar. Mawa
sugar.
flower
Yes, yes, yes.
rich in sugar.
Mm-hmm.
So that actually forms a very economically cheaper media, because major cost of this PHA production is a carbon source.
Yes, sir.
Right? Yes. So convenient.
Recently, I was thinking of it, but then I thought, okay, yes, we can produce copolymer, but again, the point comes in PHA research is alkaligens, suppose, is already accumulating more than 80-90% of its body weight
Yeah,
as
exactly.
PHA. So now the system is to upscale that. How can
Yeah,
we do the upscaling? So during my postdoc, I worked on high cell density cultivation, but with all the kind of fed batch reactors that I run, we could not exceed 30 gram per liter of biomass.
exactly.
So 30 gram per liter, and out of that, maybe the 100% purification is not possible. So 40% of the total can be the PHA.
Yeah,
So others are lost. So that was the case. So about 9 to 10 gram of the polymer we could get.
exactly.
Mhm. But there are a lot of developments in the area of PHA. Like people have made, I think in China, also previous company Metabolics is now owned by a Chinese company, and a Korean company CJ Bio is also working on this. So they have taken up some patented process. While a Chinese scientist GQ Chen, he has published a lot of paper on recombinant as well as CRISPR-based PHA production. Means CRISPR-based recombinant organism that can produce a lot of polymer using cheaper carbon source or just waste material.
Mm-hmm.
So his research publication as well as his own company is now selling PHA. So pure products they are selling to the market, purified polymer as a standards. So PHA have a lot of monomers, so their uh, thermoplastic property as well as physicochemical property varies depending on the monomeric composition of the polymer. But to characterize all such polymer, we need them in pure form. So they are selling that those standards nowadays.
Exactly.
And in Bangalore, sir, there is some startup company. I think he is Swati, if I remember her name correctly. So he is running some startup. He has established a pilot scale plant, and he is also selling our Indian product, Indian PHA standard. PHB. She is selling PHB only, not copolymers.
Very good.
PHB only she is selling. Yeah.
Yeah, field is growing as such, and
Yes, sir.
scope is there.
Yes, sir.
Yeah. So we should continue this kind of discussion. Professor Iqbal also has joined, and then he could also give some input. So it was very
Yes, sir.
similarly. Dr. Sheetal was telling about their work on cellulose production. Even some
Yes.
cellulose work is going on even in our department also from microbial source.
Okay. Bacterial cellulose.
Yes, yes.
Oh, great. So is it producing in large quantity? Because so far I know it is a slow process.
Yeah, yes,
Yes.
yes, yes.
So we need to sometimes keep it for months
Exactly.
without contamination, like batch process we have to run.
Exactly.
Mhm. Great, sir. Great to know that you are also working on PHA. You have published already, I saw. Uh,
Oh, yes, yes, you are right.
uh,
But now more activities on the probiotics,
Probiotics. Yes, I know.
AMR.
AMR also. Okay.
Yes, yes.
Great. Great. Thank you so much, sir, for
Yeah.
joining in and shedding some light. I hope if any other participant have to speak, they can come forward. Asmita Bambole, are you there? Asmita Bambole, Jayanta, are you there? You have recently joined, I guess, to this meeting. Jayanta, Ramananda, would you like to say something? Ramananda? So, yes, MN Gaming, someone has joined with the name MN Gaming. Uh, would you like to say something? Where are you from and what you are working on? So there had been a lot of discussion on various different types of microbial
Sorry, sir.
polymers. Yes,
One last question.
yes, Sarika ji. Tell me.
Sorry, sir. It is actually question out of the context. Like people here, anyone is working on the pH degradation, polyaromatic hydrocarbon degradation.
Uh, Maybe Iqbal sir, I am not sure. So those who are listening, they may come forward. Then I will tell my part. but I would like some participant to speak. So if there is some participant who has previous experience or have worked on PAH degradation, that is polyaromatic hydrocarbon, which are common pollutant generated through coal bioprocessing as well as petroleum industry. So recently we started some work of pH degradation, but we just started, so we are also learning. Let me know your question, what exactly you want to know.
You are using the different pH composition because pH is a composition of 15 different elements. So you are using the direct pH from the industries or the different components for the
Yes,
like
we worked on standard materials because we could not get some contaminated source. No, we need to characterize it.
okay, sir. So what method you are using for the isolation of the pH degrading bacteria? Like normal screening method?
Yes, usually the contaminated site, like pH contaminated site, or wherever you expect that there are some related material being dumped. You know, some industries are using some kind of pH compounds during their processing. So such effluents you can use in the from the industrial belt. You can take the sewage water also.
Okay.
Industrial area, you can get the sewage water there because they have a lot of chemicals, including these compounds. So, like in our case, we used effluents from Mathura refinery. It came, so we started screening one by one, and we got 2 3 hits, but ultimately one was showing some results, uh, and it had a good tolerance limit also to pH. So we used such organism which is tolerant as well as growing in that media. Na?
Yes. And what medium you are using, sir, for this screening method?
So we wanted to first actually follow somebody else. So we were using mineral media only. Okay, supplemented with this pH and with or without carbon source.
Okay. But the pH is normally so insoluble in water.
Yes. You are very right. So,
So how you do that?
no, so we need to make the stock, and because the organism that we are using, they produces uh, surfactant molecule. So surfactant to make them like bioavailable, and then only slowly they gets degraded. So before and after the experiment, we do the extraction process, extraction for pH, and then quantify it. So we know how much we added at zero time, and after some experiment, so after maybe for 4 days, we'll again check for the remaining amount of pH.
Okay, thank you, sir.
Yes. So before participants start leaving, there is a message in the chat box. You all should go and submit the feedback, and the certificates will be issued to you all, those who will submit the feedback. Okay. So if you are unable to see the feedback link, let me know. I will repost it in the group. So I'm sending just the link. I hope you know that this is a kind of feedback form. Thank you, Janta. I hope you guys will also be active and ask questions, because we are here only to share knowledge, and with discussion, uh, it is like shared among all of us, right? Whether you ask some small question or big question, that is not an issue, but we need to interact more. Thank you, Jayant, for joining in for today. We'll see you in next section.
Hello, sir.
Yes.
I'm studying BSc Microbiology in second year.
Mhm.
Today we are learning about virus. I have a question. Why we use minus signs in double stranded DNA or RNA replication even if we have plus signs? Because the replication needs plus signs, then why we remove plus signs and use minus signs for replication?
Mm, I did not get you. So for viral replication, why only one strand is used? That is your question. Means positive strand is used or negative. What is your question? Just a minute. Why we use, because it was out of context, so I did not take up this. Uh, and even I am not sure about the answer to this. Maybe if someone knows, please help. So far, my understanding goes, it may have to do with the kind of DNA polymerases, uh, which will initiate the process of replication. So if it is identifying the uh, negative strand or negative sense strand, then the process will follow like that. So it may have to do with the DNA replication machinery of the virus, but I am not sure about that. Maybe some other participant, Dr. Kalidos, if you may like to shed some light on this question.
Uh, I just like to share something. I don't exactly know the reason, but there are two stand. One stand engaged for the expression purpose of the protein or enzyme, and one stand involved for the just for the replication. Because in bacterial cytosol, double-stranded DNA, one stand is engaged for the expression purpose and one for the only replication purpose. So those stand we are only involved for the replication, then integrate and then replicate with the bacterial chromosome. Not exactly, but if answer is should like okay.
Yes, I think you are right correctly. So negative strand is the strand which is like an anti-sense strand. So sense strand is being followed, right? So in order to like make a copy of the genetic material, we need the exact copy. So negative strand is used to do the replication, while the positive strand is basically used to make protein synthesis, to make RNA and everything. While for DNA replication, negative sense strand is used. So, anyways, it was quite a unique question. Thanks. I think you are Faridi, or your name is not coming. So, anyways, it was a good question. Now, maybe if you come again to the meeting in the next Tuesday, or whenever this meeting will happen, we'll try to cover that. So, thank you all for joining in, and do fill the form, feedback form. Okay. All the participants who will fill the form will also be issued a certificate. Okay. And we will see you in the next meeting. I'll keep this active. You may leave the meeting once you are done with the feedback form. Thank you, Kalidas sir. I'll see you in the next meeting. It was nice to interact to you today.
Thank you. Welcome. Welcome.
Yes.
Welcome, sir.
Thank you, sir.